Hello, I'm pleased to share with you my lecture on cell biophysics. This has to do with cell dehydration during freezing. And I'll start with a motivation for this, which is related to cryopreservation or cryobiology. On the left, you see a controlled rate freezer. So this gives us the ability to freeze down materials at rates, usually tens of degrees C per minute, 1 degree C per minute, something like this. And then storage in liquid nitrogen. And you see examples of both of those. One might use this for cell and tissue banking, and some of the challenges in this area are gametes, sperm and oocyte cryopreservation, as well as primary cells, also tissues and organs. So there are a number of steps in what I'll call slow freezing cryopreservation. This is a type of cryopreservation where we allow ice to actually form in the extracellular space. There are other types of cryopreservation where we don't. An example of that is vitrification, and that'll be a topic for a different lecture. On the left, you see an example of the temperature time course of a cryopreservation with freezing protocol. And this starts with where there's a cryoprotectant that's added, and we'll talk more about CPA addition in a different lecture as well. This is like a biological antifreeze that helps the cells survive the freezing process. And then an initial cooling to a nucleation temperature in B, and then you allow the ice to form in C and sort of equilibrate prior to D, which is a controlled rate freezing step. D is where we're going to really be focusing a fair bit of our attention in today's lecture, to try and understand what are the major biophysical changes and maybe some of the injury mechanisms in that. Finally, after you're below -80, usually there's no more injury or biophysical change, and then you can put the sample in storage, like liquid nitrogen storage, which was featured in the last slide, and that would be F. And you can keep that there, and that's one of the benefits of cryopreservation and cryostorage is you can stay there essentially indefinitely prior to rapid thawing and use. So there can be potential injury at each one of these steps, and one of the big ones is in D during this controlled rate freezing, and that's what we're going to talk about. So this has been studied since back in the 1800s, and this is Ken Diller. He's a faculty at the University of Texas at Austin, who's famous for doing one of the very first cryomicroscopes that was microprocessor controlled. And he's also a history buff in cryobiology, and he's published a number of papers on Hans Mollisch's work and others, early cryobiologists. So what you see here is an example of Mollisch's cryomicroscope, and you can see in the bottom right, there is a container that allows ice or snow to be packed around the cryomicroscope, or you can imagine even putting in a slurry of ice and salt, so you could actually hit and hold temperatures. And then you could visualize your biological samples under the optical microscope. So this is described in Ken's paper, and also he's shared with me some photos. This is a more recent generation. This was in the late 1980s. This was the first cryomicroscope I used in my lab, and this is actually from Maury Kosman's work at MIT. He was a graduate of Ernie Cravallo, and so was actually Ken Diller at MIT. And you can achieve rates of up to about 400 C per minute with this. This is an optically transparent convective cryomicroscope. And some of the key features are this chilled nitrogen gas that flows underneath an indium tin oxide deposited window. On top of that, so that indium tin oxide deposited window is like a heater. And there's a sapphire window that holds the sample. So this is optically, all of it's optically transparent. There's a foil thermocouple below the sapphire disc, and that allows for feedback control with a microprocessor or computer. so that you can have differential cooling or heating depending on whether you're trying to cool down or warm up your sample. So this is published in Cryoletters, and it was a very, at the time, sort of state-of-the-art instrument. Since that time, Lincoln Scientific makes probably, I would say, arguably the best cryomicroscope in the world, and this is being used now by a lot of groups. And I call it a conductive cryomicroscope. There's a metal disc here through which you have liquid nitrogen or gaseous nitrogen flow. You have a platinum RDT sensor and heater that's embedded into the metal post. There's also an optical window here, this hole that you see, and you can clamp down with an O-ring this top so you don't get any condensation, or you limit the condensation. You can also do other things like evacuate the chamber, and you can freeze dry this, the system while you're keeping it at liquid nitrogen temperatures. So this is a very nice, very robust stage. In our hands, it goes at about 130 C per minute. They claim that it can go up to 5,000 C per minute. I think this is really what they call quench cooling, when they essentially turn the heater off. If you've got the heater going at max, you know, holding it up close to, let's say, 0 degrees or minus a couple degrees Celsius, and then you just drop, let the bottom go out, you can reach something close to 5,000, but I don't believe that that's a controlled rate. So what does this look like? These are some examples, and these, don't take these scale bars too seriously, but these mouse oocytes are much larger. They're 100 microns, and the cancer cells are more like 10 to 15. So this is a little bit off, but you can tell there's an order of magnitude difference in size. The mouse oocyte here, this is something I took off of a website from John Morris's group, Asmatote, which was recently purchased by GE a couple of years ago. So you can see the ice with cryomicroscopy. You can watch the ice growing into the solution around the cell, and the cell begins to dehydrate. So you can begin to see some invagination and some changes in the membrane here. And this is a kind of a slow process. And partly that's because it's a slow freezing process. Partly it's because this is a huge mouse oocyte with a relatively low permeability. If you look at these cancer cells over here, and they're going at a faster rate of cooling, you can see that the dehydration is much, much faster. And if you go at a little bit faster rate with those LN cap cells, these are cancer cells, you begin to see this darkening effect here. And this has to do with ice actually forming inside the cells. So it's not just water transport, but you're competing. There's another process here where the water within the cell is supercooling to the point where it actually forms ice. That happens if you trap enough water or you go at a fast enough rate on the way down. We're not going to talk about IIF here, but we'll talk about it, intracellular ice formation, in another lecture. So this is a cartoon. This was actually put together by David Smith during his PhD in my lab. And what you see is a cell originally unfrozen in solution. And as the ice forms, the ice is pure water, and it begins to concentrate the solutes, which are these little orange particles. If you go slow, the ice has time to grow around the cell, and the concentration of the solute rises, and you're extracting water and you're dehydrating the cell. If you go faster, like what I showed in that last video, you can sometimes trap enough water and you supercool it, and it actually nucleates inside the cell. That's called intracellular ice formation. Again, we won't really be getting into that today. Peter Mazur really is the one who talked about these competing biophysical processes, and is considered sort of the grandfather of the field. And so what he pointed out in his American Journal of Physiology paper is that actually, if you look at survival versus cooling rate, there's sort of like this optimal rate for every type of cell that you work with, and it can go over a variety of logs of cooling velocity or rate, degrees C per minute. And that relates to the size and the permeability of the cells and a variety of things that we're going to get into here shortly. But this is really what we're interested in as cryobiologists, is like, what is that sweet spot? What's the best rate to cool down the cell of interest so that we can preserve it? And it turns out that Mazur pointed out that there's basically the survival is a competition between avoidance of two major types of injury: slow cooling injury when you dehydrate too much, or intracellular ice formation when the water is trapped inside the cell. And he calls this an inverted U curve, and for obvious reasons. And it's really just a competition between dehydration and intracellular ice formation. He calls this a two-factor hypothesis. He originally published that in 1963, but it's very nicely reviewed in his MJ Phys article in 1984. So as engineers, we started looking at this. This is actually work from John McGrath. And you can see that, the cell, if you start at the first order, thinking about it as kind of a bag of salt water with a semi-permeable membrane, you have pure ice growing into this solution. And you have water and solutes and ice. The solute concentration is going to go up, as we talked about, and that's going to tend to reduce the volume of the cell. And so this is what you see over here is y-axis normalized cell volume. As the temperature drops and more ice comes into the system, you're dropping dramatically your volume. However, as you go up in rate to 10, 100, or 1,000 C per minute, you trap more and more water in the cell. And eventually, as you go to a low enough temperature, you will get that intracellular ice formation. If we think about what's actually the ideal rate, it's probably someplace that's just a little bit off this equilibrium curve, and maybe it only has 5 or 10%. Peter Mazur has speculated, something in that neighborhood of cell volume, so that whatever might happen with ice would be innocuous in the cell. If you have a huge amount of ice in there, it's obviously going to be damaging. This is from his, John McGrath's chapter in Heat Transfer in Medicine and Biology in 1985. So this is a really simple model for this. We basically look at the intracellular compartment as idealized solution, just solutes in water. And outside, we have osmotic pressure that's being driven by the salt being kicked out from these pure ice crystals that are forming. And then that osmotic pressure difference is driving a flux of water. And it's going through a semi-permeable membrane with a membrane hydraulic permeability. So this is, again, hydraulic permeability. John studied this for his PhD. He wrote a very nice treatise on this back in 1988. And really, these were some of the first, studies that were being done on how water moves through the lipid bilayer. This is transition state theory. There are a number of others, kink models and so on, that have to do with the energetics of how water moves through the lipid bilayer to move from one side of the membrane to the other. From our point of view, it's passive. All active transport is basically shut down because it's at low temperature, and it's potentially there's some facilitated transport, like with aquaporins that have been found in red blood cells and sperm, but all of that will be lumped into an overall hydraulic permeability. So how do we understand the extracellular solution and the osmotic pressure and the osmolality concentration that is arising because of the ice forming? So you can think about this from the point of view of freezing point osmometry. This is a really simple concept. Basically, if you take pure water in blue and you put it into an osmometer, you'll drop the temperature with this cooling block, and eventually you can trigger freezing initiation. The temperature will basically rise to the phase change temperature of the solution, and then it'll drop back down. So for water, that's 0 degrees, but if you put saline in there and you trigger freeze initiation, it'll never make it back to 0 degrees. It'll be at some temperature that's actually a temperature that's related to the concentration. And so you can see this basic, temperature versus concentration plot here for salinity. And this is the basic concept of freezing point osmometry. And You can actually see this in a little more detail if you go into a binary phase diagram. In fact, this is a phase diagram in some sense, what you're getting from freezing point osmometry. You can also get it with even more detail from differential scanning calorimetry, where you would have a sample of a couple of milligrams of solution of saline of different concentrations, and then a reference pan, and you heat it up or cool it down, and you can monitor the amount of energy that goes into a phase transition. And you can get information like this on the left, where you can actually see eutectics, which is a secondary phase, solid phase transition, down at about minus 21.1. And you have more and more of that eutectic depending on how much concentration you have of salt in your solution to start with. And you can also see where the melt is here, in terms of this is a heat release due to melting of the salt solution. So that allows you, this type of information allows you to put together an even more definitive phase diagram. From our point of view, we just want to make a concentration versus temperature link, so that we can begin to look at this model in a more careful way. And so it's really, temperature versus concentration phase diagram. You can actually get, by integrating Gibbs-Helmholtz, you can actually get to the chemical activity of water at any temperature, which relates back to concentration or to osmotic pressure. And that's what this relationship is here. You can also see, this is just from Wikipedia, if you just have a U, manometer here with a semi-permeable membrane, and you add concentration of a solute on one side, it'll drive flow through the semi-permeable membrane, and you'll get a change in the height. That change is basically the hydrostatic pressure that's basically being it is balanced by the osmotic pressure from the solute that you've added. So basically, concentration drives flow. If you have a higher concentration outside the cell, it's going to pull the water, just as you see here in this example. So this is what the transport equation looks like. Basically, flow, flow out of the cell due to this osmotic pressure difference between the internal and the external parts of the cell, and chemical activity, chemical potential, and chemical activity of the two intracellular and extracellular spaces. So the only thing left here is to talk about the chemical activity inside the cell, which, if we assume it's ideal, is just the mole fraction. And we make all of these assumptions about ideality, and we also recognize that part of the cell is actually what we call non-solvent. It's just part, it's solids that are not participating in the osmosis or the water movement. So we can also ask, is this well-mixed assumption really a good one? And this Peclet number, which is really diffusion versus convection, is a way of looking at that. It's kind of like a Bio number analog, if you will. So you have convection on the top over diffusion on the bottom. And if you take characteristic numbers like the velocity, 0.01 micrometer per second would be the velocity you could get from like a hepatocyte efflux, and characteristic size of a cell and volume diffusivity of solutes in the cytoplasm, and you put all that together, you get these very low numbers, which suggests basically that diffusion dominates convection, how much water would be leaving the cell membrane, and therefore you are fine in making this well-mixed assumption. There's a whole bunch of other assumptions that are listed here. We assume that the membrane hydraulic permeability is temperature dependent. Extracellular solution is in equilibrium. That's basically the phase diagram assumption. We haven't talked about this yet, but the hydraulic permeability will be concentration dependent, but this is really only if a CPA is present. And that CPA might be, like DMSO or a glycerol, one of these antifreeze, biologically compatible antifreeze molecules. There's also an assumption of the surface area of the membrane being constant. We're assuming only water is moving through the semi-permeable membrane, no solute, and that the cell and the environmental temperatures are the same, the intracellular solution is ideal, and that the heat of fusion is constant. And this is again covered in John's chapter in heat transfer in medicine and biology. And as you can see in this table over here, there was a lot of work that was done on this that he compiled for that nice review. So the temperature dependence of the permeability was actually, so you can see the whole, this is the whole equation. It's the volumetric change with temperature now. B is actually the rate of cooling. So if you're cooling very, very fast, you can see that's in the denominator. You're not going to get a lot of volumetric change. And then the rest of this, these are the chemical activities that we just derived and talked about in the last slides. Finally, this membrane permeability is this Arrhenius relationship that Levin was the first to propose. And so as you go to a lower and lower temperature, you're basically shutting off the permeability, and you'll shut it off more in a more extreme way with the higher the activation energy. So what does that look like for an example of data fitting? Here's some human hepatocytes. You've got ice crystals growing in, -1, -2, -3, and -5, and you can see that they are shrinking. And if we make the assumption that they're essentially, when you're doing this analysis, you take the area to be representative of a volume. So you can do basically a relationship of the areas to the volumes with a 3/2 power assumption. And then you can get volume here, normalized volume on the y-axis, temperature and degree C on the x, and you can actually get the dehydration behavior of the cells. And then you fit it to the equations that Ron Levin and others have worked with, and you can extract these parameters. So the membrane reference hydraulic permeability, usually at the reference temperature, which is usually the phase change temperature. ELP would be the activation energy for the process, and then the initial and the non-solvent volume. This is something we published back in 1995, the specific data set, but Mazur and Levin did many things like this and others in the past. The impact of CPA on water transport and permeability is something that a number of people have looked at, including us. You can see here some of our data on the left. You have an increase from 0 to 1 to 2 molar DMSO. This is a cryoprotective agent. And then you have rates of 5, 10, and 50 C per minute. And you have this dehydration response, normalized volume, and then again, temperature on the X. And as you go up from 0 to 1 to 2 molar, you can see more and more water is being trapped as you go to lower temperatures at the same rate. And as you go up in rate, as I already showed you with John McGrath's work, you're trapping more water as well. So you're basically out competing. That's because of this B in the denominator here. As this B goes up, there's less time for this volume to change. And if you do all the fitting, you can show very clearly that the activation energy reduces, and the reference permeability for hydraulic permeability also reduces with molarity. And so this was published in Journal of Biomechanical Engineering back in '98. Here's some just reference from the literature looking at a variety of different, it's natural log of LPG reference permeability to the activation energy. And you can see there seems like maybe there is a bit of a relationship, but I don't think anyone has completely worked that out. There is a lot of interest in understanding a little more mechanistically, though, what is the permeability and how does it relate to, let's say, lipids? And so Wim Volkers has been doing a lot of work in this area using Fourier transform infrared spectrometry. And what he's shown is that lipid hydration actually mirrors cellular hydration. So what you see here, if you freeze rapidly or slowly, you get a very different behavior in the lipids in terms of the phase change. And that's shown in this BBA article from Wim. And so if you actually nucleate at -10, you get a very rapid intracellular ice formation, which keeps you in this more fluid gel. If you go with nucleating at -3, it's a much more dehydration slower case, and -6 is sort of in between. So you're looking at lipid packing at -3 and lipids staying very open and more fluid at -10. And you can actually see that there's basically, this is tracking with the behavior of the dehydration of the whole cell. So we looked at that a little bit more carefully, and we looked at the activation energy for this packing. And what we found in 3 different cell types is that the activation energy by cryomicroscopy, when you're watching the cells actually, dehydrate, as I've shown, or you're looking with FTIR at their packing based on, how they respond in freezing, that they're very similar. And so it seems that, this mechanistically, what's going on when you're shutting down the water transport, and this sort of makes sense, is that there's just less room for the water to move through these packed lipids versus the case where it's more open. You can use this in sperm cryopreservation, and the reason for that is that these sperm are really, really small, so it's very hard to look at this with cryomicroscopy. These are only like 1 to 2 microns across, and you really can't see the shrinkage of a sperm during cryopreservation in the presence of ice. So using either differential scanning calorimetry or Fourier transform infrared spectrometry, you can get this indirect information about water transport and permeability. And the reason why that was important is that a lot of the other, so people were making measurements with Coulter counters and things up in 20-degree range rather than in the presence of ice. And you're off by several orders of magnitude in terms of your prediction of what the permeability should be. So Wim and others have been using these types of measurements, Ram Devareddy at LSU, to actually do a better job predicting where the optimal rate should be for sperm versus the superzero simulation. So this is just an example of how FTIR could be used. It also relates a little bit to cell injury. So now that we've learned that there's this biophysics of dehydration and intracellular ice formation, the next layer is really like, how does that all, like where are the injury mechanisms then to the lipids and the proteins that would lead to cell death? Of course, ultimately we want to avoid those so that we can have preservation. And, so some study of lipid phase changes. This is from Wolf and Bryant. This is an area where there's been a fair bit of work and interest. And one very dramatic example of injury is from Peter Stepankas's work. This was in plant protoplasts, where he showed that you can actually get an endomembrane coming in contact with the plasma membrane and then getting inverted hex 2 formation during freezing. And then upon thawing, you basically have, you're exuding the content. Or you could have, the plasma membrane coming together and basically fusing. And then when the cell wants to completely come back to its normal volume and the surface area of the membrane, there's not enough surface area left, and you get what's called expansion-induced lysis. So Peter was the first to really talk about this as a lipid mechanism of lipid injury during dehydration. So in conclusion, we showed that freezing induces crystallization, which increases extracellular concentration of solutes, and that leads to an osmotic pressure, which drives water out of cells. Dehydration can be measured by cryomicroscopy and modeled with a 2-compartment model. The amount of dehydration depends on the membrane permeability of the cell, the rate of cooling, and the presence of CPA in the solution. And the amount of cellular dehydration is important, as the fastest rate without significant intracellular ice formation is typically the highest survival for the cell type. And cellular dehydration is linked to membrane dehydration and permeability drop. That was that work from Vim. And too much dehydration can be detrimental. So you can have these lipid phase changes and actual mechanisms of injury because of it. So just some references to think about if you want to take any of this further. There's some historical work in sperm. Some say that this was like the paper that kind of started the field of cryobiology. That's not probably entirely true, but this has gotten a lot of interest. Polgy, Smith, and Parks, where they did revival of spermatozoa after vitrification and dehydration at low temperatures in nature. There's a whole bunch of DSE and FTIR studies also on sperm that are related to, Vim's work and Ram Devareddy's work and others that have worked with both DSE and FTIR to use mechanistic studies to do a better job with sperm cryopreservation. There's also some nice background in terms of what I presented, measurement of water transport in cells, Gopert and the early cryobiologists, Peter Mazur's work in the Journal of General Physiology, the two-factor hypothesis, Ron Levin's work, already mentioned, for Arrhenius permeability modeling, McCaw and Diller showing some cryoprotective agent concentration dependence of LP, and our work, which was David's. There's also some nice reviews of measurement and equipment by Ken Diller and John McGrath, both published in Cryobiology in 1997. And a separate topic for another day is how do we extend this to measurement in tissues, because tissue-based cryopreservation is also a huge issue, an increasingly important issue. Some homework thoughts that we can get into. How do we use the Gibbs Helmholtz to actually get to osmolality and to understand the extracellular concentration? How about water transport modeling? And we can do this with software. How do we predict the fastest rates before intracellular ice formation to actually get the best rates for sperm, as an example? Equilibrium cell volume concentrations to basically learn to use intracellular and extracellular activity and concentrations to understand how, what volume a cell will actually have at equilibrium at a given temperature. Boyle-Van't-Hoff behavior, which gives you your non-solvent volume, phase diagram calculations, and then maybe some Peclet number calculations. So with that, I'll thank you all for your attention, and I hope this was helpful.